you may not anticipate gene sequencing, genome sequencing, being involved in diplomacy. And I would like to show, if I may, if you can turn on the, okay. This simply is a mechanism, this diagram, to show that life and the problems are highly integrated and complex. Biology really is biocomplexity rather than just biology. And it requires integrating information from the cellular level to the community to perhaps even the cosmic level. And that it is important to understand that we are connected continent to continent, because as this particular NASA slide shows, there are these dust storms that sweep from the Sahara to Western Europe, from the Gobi Desert to the United States, and certainly within Asia, so that we are connected whether we like it or not, and that these connections, as shown by this particular one of the Saharan dust storm over Europe, carries many, many microorganisms. These are the ones that can be grown. There are many that are present that can't be grown, viruses and bacteria. We're connected continent to continent biologically. I'm going to go through quickly because I want to, let me just point out that in seawater, for just 1 teaspoon of seawater, the diversity is extraordinary. And here in Malaysia, you have one of the richest coral reefs in the entire world. You also have a diversity that's unmatched. We have not yet harvested that diversity, and I think we need to have a joint effort where this diversity can be decoded and the genetic capacity utilized without destroying the resource itself. That is what the new biotechnology allows us to do. The microorganisms range from this Namibian isolate that can be seen with the naked eye, it's a bacterium, to the smallest viruses which have to be observed with an electron microscope, and they're so small that they carry only a fraction of a genome that one would expect. They don't carry a whole chromosome. Let me just go quickly to point out that through genomics, we're getting a better understanding of microorganisms. You've heard about the E. coli that has created havoc in Germany. In Spain, has wrecked practically the agricultural economy of Spain because it was claimed to be an E. coli that came from vegetables in Spain. It turned out not to be the case. Again, diplomacy and understanding health sources. But let me just point out that you see 2 circles, actually 3 circles. These are the different kinds of E. coli. There are the safe kind that are in our intestine, all of us, that provide vitamins for us. And then there are those that pick up genes that convey the capacity to cause disease. And it is the ability to monitor those fragments of DNA that are picked up that can turn a normal pacifist bacterium, if you will, into an aggressor and a disease agent. I'm going to go quickly because I want to get to showing you a bit about, I'm sorry about this, but I didn't have time to cut it down. I'm just showing here how bacteria, just as we humans, have an enormous amount of diversity. This is the bacterium at the top that causes cholera. I know you have sporadic outbreaks in Malaysia. It's not a major disease as dengue to be feared, but it does appear. In any case, the bacterium, when it attaches to the intestine, as you can see in the lower left, produces a toxin that causes the manifestation of a disease. We now know, because we sequenced it, and it has 2 chromosomes, and we now have been able to determine genes on this bacterium that causes the massive epidemics of cholera. Recent studies, we took historical strains, recent strains, strains from Asia, strains from Africa, strains from the US, and we were able to demonstrate 2 important things. The red lines across the top and across the middle. The top represents 1 chromosome of cholera. The bottom represents the other chromosome. I don't think this has a pointer. In any case, comparing strains, sort of pulling the chromosome, stretching it out, and comparing them, we can see that, very good, that's great, that's perfect. Yeah. We can see that these strains are all, strains are cholera strains. And, let me go back, sorry, I'm just learning this technological device here. These strains are cholera strains. This is that reference strain, the first one that was sequenced. This is the 2nd chromosome. This is the 1st chromosome. Very interestingly, we find that there's a great deal of difference amongst strains of cholera from different parts of the world. These are other species, and you can see blue means the genes are missing. So you can see a great deal of difference between other species. Now let me go very quickly to the Haiti analysis. This is a whole lot of more data than I hope I'm not boring you too much, but let me get to my final point. One sidebar to make here is that one of the discoveries we made was that in a sample collected from a hydrothermal vent 2 miles beneath the sea, we discovered that there is a cholera bacterium that is very similar to the ones that cause disease, which means we can trace the origin of this bacterium perhaps to the very deep sea. Let me get now to the Haiti study. I'm moving very rapidly here. Can you, yeah. We found just the deep sea strain has pathogenicity genes just like the epidemic strains. We have also discovered that in a country like Iceland, never had cholera, but where the geothermally heated waters are located, sampling being done by a former student, we found that indeed there is a cholera bacterium at these sites. In other words, we can see the bacteria. These are cholera strains at these different sites that are geothermally heated. And let me now go to the cholera data. Okay. The point I want to make, let me back up. What I want to make that's very dramatic is that there was an earthquake, as you know, devastating, in January last year in Haiti. Severe disruption of the ecosystem and certainly the people. Then, about August, September, there was a huge hurricane that went through, created flooding. And as if the poor people of Haiti hadn't suffered enough, there was this massive cholera epidemic. And some preliminary analyses were done, and the conclusion was very quickly and too rapidly concluded that it was identical to an Asian strain. And the charge was made that cholera was brought in by the peacekeepers. There were riots. There were deaths during the riots. The claim was that Nepal peacekeepers were the source of the cholera. Now, we were able to, through working with a physician on the ground in Haiti, to obtain specimens from 60 different cholera patients all over the country. And we were able to sequence the cholera strains. And we found that, in fact, let me go back, a little too hasty on this. We found that, in fact, when you did the full sequence and did the comparisons, strain by strain by strain, against the Haitian strains, and the genes that could be transferred back and forth were removed from comparison and just looked at the backbone of the strains, we found that they are different. These are Haitian strains, and this is the so-called Asian strain relative. It's quite separate, and we've submitted a manuscript to Science that's under review at the moment, suggesting that it's far too premature to have ascribed the cause of the Haitian epidemic to the Nepal peacekeepers. Now, this is very high technology science, but you, as ambassadors going out to other countries, are going to be faced with incidences that are equally science-based. And you do need input. You do need the capacity to have this information, because had there not been this rush to judgment, there might not have been some people who died. They might not have died. So this is a very, how shall I say, very current, a very relevant example of the role that science plays in diplomacy. It's the understanding of what is happening in the country in which you are located, and the interactions of these events in other countries, and being able to have a global view, a global interaction, and the ability to make decisions that are evidence-based. A very important message. The diversity of these strains are quite substantial. Their metabolic functions are very different. The conclusion is that we need to incorporate the cutting-edge science. We need to understand that science, engineering, and technology discoveries are moving at an incredible pace, and it's almost head-spinning in the speed with which change is occurring. But that means you must make your ties to the scientists and the engineers and the technologists, so that the challenges which faces the world today indeed are formidable, but the solutions to the